Transposable element. In this chapter, we will examine the actions of bacterial transposable elements, including their mechanisms of transposition and reintegration into host DNA. We will also distinguish between RNA-dependent and RNA-independent eukaryotic transposable elements. Finally, we will evaluate the homology between transposable elements and viruses to understand their evolutionary origins. Virtually all organisms contain mobile DNA sequences known as transposable elements, or transposons. The movement of these elements is called transposition. Due to the potential disruption of essential genes and induction of harmful rearrangements, there is considerable selective pressure to keep transposition frequency low. While transposons have been studied extensively in bacteria, they are present across all domains of life. Let's begin by focusing on bacterial transposable elements. These can be categorized into simple and composite forms. A common example of a simple element is the insertion sequence, or is element. These short DNA sequences can insert themselves into various positions in the genome and are typically the most basic form of transposon in bacteria.
Transposons are segments of DNA that are capable of shifting from one location to another. A transposon enters the cell by being carried on a plasmid. A transposon can then move from the plasmid into the host cell genome. A transposon can move from one site on the host genome to another site on the host genome. When some transposons move, they replicate, leaving a copy in the original position. A transposon can also move from the host genome to a plasmid.
Take the IS1 element, for example. It is a 770 base pair sequence with imperfect 30 base pair terminal inverted repeats. Within these repeats lies the TNP gene, which encodes for a protein known as transposase. This enzyme facilitates the cutting and reinsertion of the element into host DNA, a fundamental step in transposition. Composite transposons contain two elements flanking one or more functional genes. Over time, one of the flanking transposase genes may become non-functional. The genes between these elements often include those conferring antibiotic resistance or enabling carbon source metabolism, making them clinically significant. Examples of composite transposons include TN5 and TN10. These transposons have as elements designated as IS50L and IS50R for TN5 and IS10L and IS10R for TN10. Each element has outer ends O and inner ends, i.e. the transposons often carry antibiotic resistance genes, and the TNP gene encodes transposase, essential for movement. R equal purine, Y equal pyrimidine, W equal O or T, N equal any nucleotide. Let's walk through the process. First, the transposase binds the outer ends of these elements. It then cuts at these sites, generating blunt ends. A double-strand break is introduced at the target site in the host DNA. The transposon is then inserted, and the host DNA repair enzymes fill in the gaps and seal the nicks. This figure illustrates Hairpin-mediated transposition, where a loop DNA structure forms during cleavage. The process is asymmetrical and highly coordinated, allowing the transposon to jump efficiently while minimizing genome instability. This form of movement is known as cut-and-paste transposition.
Transposable elements are discrete DNA segments that can move from one site in DNA to another site. A DNA cut-and-paste transposable element moves by excision, followed by integration. Transposition begins when the transposase binds to terminal inverted repeat sequences at each end of the transposon. The transposase molecules then pair, bringing together the transposon ends and activating the transposase activity. The transposase then excises the element by generating double-strand breaks in the DNA at each end of the element. The transposase then directly joins the transposon ends to the target site in the target DNA. Note that the positions of strand joining are staggered on the top and bottom strands of the target DNA, such that the ends are flanked by short single-strand gaps. These gaps are then repaired by host repair enzymes to regenerate duplex DNA. This staggered target joining and gap repair results in duplication of the target sequence, a feature characteristic of the insertion of all transposable elements. Excision of the transposon leaves a gap in the donor DNA which must be repaired to regenerate an intact DNA molecule.
An interesting regulatory mechanism involves GATC methylation. After replication, the DNA is hemimethylated, the old strand is methylated, and the new is not. This transient state activates transposase expression. Right after the replication fork passes, TN5 can jump ahead, capitalizing on the timing of methylation to enhance its mobility. Unlike TN5, the TN3 transposon uses a replicative mechanism, meaning it copies itself to a new site while leaving the original copy behind. TN3 is a 4,957 base pair element with 38 base pair inverted repeats and contains the TNP transposase, the res resolvus, and an ampicillin resistance gene. This gene combination is especially concerning in the spread of antibiotic resistance. The process begins with transposase nicking the donor DNA. Then, DNA polymerase extends the three ends of the target DNA, forming a cointegrate structure. Next, resolvus facilitates recombination between two resolution sites, separating the two elements and completing the process. The result is a copy of the transposon at both the original and the new site. So what are the two major types of bacterial transposable elements? The correct terms are cut and paste and copy and paste mechanisms. These mechanisms differ in whether the element is excised and moved or simply duplicated into a new location. Transposons have found a critical role in biotechnology. A transposon with its own transposase gene is called autonomous. In contrast, a synthetic or engineered transposon without a transposase gene is non-autonomous and requires the presence of a helper element to mobilize. These tools are extremely useful for mutagenesis and gene function studies in bacteria. Why are transposons useful for functional genetics? because disrupting a gene allows researchers to observe the phenotypic consequences, shedding light on the gene's role. Likewise, disrupting regulatory regions can help identify important DNA sequences involved in gene expression. Thus, the answer is all of the above. Eukaryotic transposable elements fall into two main classes. Class I transposable elements require an RNA intermediate to transpose. These are known as retrotransposons. In contrast, Class II elements use a DNA-only mechanism, typically via cut and paste transposition. Both classes can exist in autonomous and non-autonomous forms depending on whether they encode the necessary transposition enzymes. The HAT transposons are DNA-only transposons named after three founding members: Homo in Drosophila, AC in maize, and TAM3 in snapdragon. These elements operate via a cut and paste mechanism, similar to bacterial TEs elements, but with a few key differences. For instance, studies on the Hermes transposase in houseflies helped elucidate the mechanism, which involves formation of a hairpin structure on the donor DNA. This figure outlines the transposition mechanism used by Hermes, representative of the HAT family. The DNA-only transposase cuts and mobilizes the element, generating a hairpin intermediate, which is resolved upon integration into the target site. This mechanism differs from bacterial transposons, which typically form hairpins on the transposon itself rather than on the donor DNA. Interestingly, antibody and T-cell receptor genes are formed by a mechanism that resembles Hermes transposition. During lymphocyte development, these genes undergo site-specific recombination, generating immense diversity. This allows our immune system to recognize up to -0 to the power 1 unique antigens, highlighting an evolutionary link between transposition and adaptive immunity. Ig heavy chain genes consist of repeated DNA segments labeled V for variable, D for diversity, and J for joining. These segments are joined to form a functional gene that is then linked to a constant region. Similarly, light chain genes use V and J segments. This recombination brings previously separated gene fragments together, enabling the production of a wide array of antibodies. There are three key contributors to immune diversity. First is combinatorial diversity, where different V, D, and J segments are randomly selected. Second is junctional diversity, which arises due to imprecise joining. Finally, pairing diversity occurs when different heavy and light chains combine. This diversity arises through VDJ recombination, which involves programmed double-strand breaks in DNA. This figure depicts how V, D, and J segments are rearranged to produce a functional immunoglobulin gene. Initially, these segments are separated, but recombination joins them to a constant region. This rearrangement is essential to generating functional antibodies capable of recognizing diverse antigens. Each V, D, or J segment is flanked by a recombination signal sequence, or RSS. These sequences serve as recognition sites for two key enzymes, RAG1 and RAG2. These enzymes are expressed only during lymphocyte development, underscoring the controlled and developmental nature of this site-specific recombination process. The Sleeping Beauty transposon is a reactivated DNA transposon originally found in fish. In 2014, researchers used site-directed mutagenesis to restore its functionality, creating a synthetic non-autonomous element capable of transposing in human cells when provided with a transposase. This development offered a powerful tool for gene therapy and functional genomics in mammals. A historical note: Barbara McClintock discovered transposable elements in maize during the mid-20th century. Her pioneering work on genetic mobility earned her the Nobel Prize in Physiology or Medicine in 1983, and she remains a central figure in molecular genetics. Now we shift to class I transposons, also known as retrotransposons. These elements copy an RNA intermediate and generate cDNA through reverse transcription. The resulting DNA is then integrated into the genome using a cut and paste mechanism. Retrotransposons are widespread in eukaryotes and share features with retroviruses. In humans, they make up roughly 8% of the genome, although most are inactive. Retrotransposons can resemble retroviruses. A typical LTR retrotransposon has long terminal repeats flanking genes such as Pol and GAG. The Pol gene encodes reverse transcriptase, integrase, and protease, while GAG encodes structural proteins. Unlike retroviruses, retrotransposons lack the ENV gene, meaning they cannot form infectious particles. LTR retrotransposition starts with transcription from the LTR promoter. The RNA transcript moves into the cytoplasm where Pol genes are translated into enzymes including reverse transcriptase and integrase. The RNA is then reverse transcribed into DNA. This DNA enzyme complex enters the nucleus and integrates into the genome via a cut and paste mechanism. There are two major types of non-LTR retrotransposons: lines and signs. Lines, or long interspersed nucleotide elements, are usually 5 to 7 kilobase pairs long. Signs, or short interspersed nucleotide elements, are typically less than 500 base pairs. Together, these make up the most abundant transposable elements in the human genome, with over 1 million insertions of each. Line elements transpose using a mechanism called target-primed reverse transcription. The line is transcribed and translated into endonuclease and reverse transcriptase. The endonuclease makes a nick in the target DNA, and reverse transcriptase uses the line RNA to extend the 3' end of the nicked site, beginning the process of DNA synthesis. In the second step, the endonuclease nicks the opposite strand, and reverse transcriptase continues copying the RNA into DNA. The RNA is displaced and degraded, and DNA ligase seals the newly synthesized DNA into the genome. LINE1 elements in humans retain the ability to autonomously transpose, though this occurs very rarely. Signs, while similar to lines in structure, do not encode endonuclease or reverse transcriptase. As non-autonomous retroelements, signs rely on line-encoded proteins for mobility. The most common sign in the human genome is the ALU element. Signs are highly valuable in population genetics, forensics, and as markers of evolutionary history. Interestingly, the LINE1 machinery can sometimes reverse transcribe cellular mRNAs, creating process pseudogenes. These pseudogenes lack introns and regulatory elements and are usually non-functional. However, their presence provides independent genomic evidence of evolutionary processes and gene duplication events. Some eukaryotic transposons are nearly identical to retroviruses and may have given rise to, or evolved from, viral ancestors. These retroviral-like transposons often contain LTRs, GAG, and Pol genes, and may even include vestigial versions of ENV. Their evolutionary implications continue to be explored in genomics research.
Retroviruses are viruses that infect eukaryotic cells. Retroviral virus particles are also called virions. They contain 2 RNA copies of their genome as well as viral proteins, all inside a protein shell called the capsid. The retroviral capsid is surrounded by an envelope derived from host membranes that also contains viral glycoproteins. Let's now consider the retroviral life cycle in detail. During infection of a cell, the virion binds to a specific surface receptor protein via the viral glycoproteins. The viral envelope then fuses with the host cell membrane, delivering the capsid and its contents into the cell. Once inside the host cell, the viral RNA genome is converted into a double-stranded RNA genome by the virally encoded reverse transcriptase, an RNA-dependent DNA polymerase. This viral DNA genome is also called a cDNA. The termini of the viral DNA genome are directly oriented repeat sequences called long terminal repeats, which are several hundred base pairs long. The viral integrase interacts with the ends of the viral DNA. The capsid is then disassembled and the cDNA integrase complex is transported into the nucleus. The viral DNA is integrated into the host's genome in a transposition reaction mediated by integrase. Once integrated, the viral DNA is called a provirus. The provirus is transcribed by the host's RNA polymerase, forming mRNAs, which are translocated into the cytoplasm. Copies of these mRNAs will also be packaged into new virions. Once in the host cytoplasm, the viral mRNAs are translated by the host machinery to yield the capsid proteins, membrane glycoproteins, reverse transcriptase, and integrase. The protein components of the capsid then assemble, encompassing the RNA viral genomes and the viral proteins. Viral glycoproteins then assemble in the host membrane, forming sites of capsid interaction. Membrane-enveloped virions then bud from the cell, ready to infect a new cell. Retroviral-like transposons are elements whose intracellular life cycle is very similar to that of retroviruses, but which do not exit the host cell. As a result, they are not transmitted from cell to cell. Like a retrovirus, the integrated form of the retroviral-like transposon has direct repeats called long terminal repeats, which are several hundred base pairs long. The provirus is transcribed by host RNA polymerase to generate mRNAs. These mRNAs are transported to the cytoplasm. Once in the cytoplasm, the mRNAs are translated to generate viral proteins. The capsid then assembles, packaging the viral RNA and proteins and forming a retroviral-like particle which does not exit the cell. The RNA is then reverse transcribed to form the retroviral-like DNA, also called a cDNA. The integrase then binds to the long terminal repeats of the cDNA. The cDNA with its bound integrase is transported into the nucleus and is then integrated into the host genome to form the provirus, completing the cycle.
This slide provides visual references from landmark papers by Malik and Eichbusch 2001 and Flavel 1999. These studies catalog the diversity of retrotransposons across eukaryotes and emphasize their structural and functional similarities to viruses. In summary, eukaryotic transposable elements are diverse and influential components of the genome. From cut-and-paste DNA transposons like HAT and Sleeping Beauty, to RNA-mediated retrotransposons like lines, signs, and LTR elements, they represent an evolutionary force that shapes gene regulation, genome architecture, and immune system diversity.
There are sections of an organism's genome, called transposons, that have no other function except for inserting copies of themselves elsewhere on the genome. And there are many very well-known sequences that do this. Two such sequences are signs, or short interspersed transposable elements, and lines, or long interspersed transposable elements. There are about 850,000 lines and 1.5 million signs scattered throughout your genome, accounting for nearly 30% of the entire sequence. While they are useless to the genome, and sometimes cause significant damage, they are useful to our investigations, since essentially the only way for them to go from one organism to another is through direct DNA duplication and inheritance. Your lines and signs are given to your children. The parts of your DNA that make up your genes are relatively small sections scattered among other useful parts of your genome, as well as your lines and signs. Like fingerprints, the patterns recognizable in these non-gene sections are unique to individuals. They are similar in relatives and less similar in distant relatives. This is the basis of DNA fingerprinting. And since these sequences are passed down from parent to child, finding the same sequence in the same place in two different organisms is direct evidence that the two organisms have a common ancestor. Biologists have used this idea to demonstrate exactly how different species are genetically related. Here are two examples. 3 different identical sign sequences are found in the same genome location of whales and hippos, which are closely related, while camels and pigs, which are not closely related to whales and hippos, do not have the same sequence. The most numerous sign element in your genome is called the ALU. In the human alpha-globin cluster, there are 7 ALU elements, and each one is shared with chimpanzees in the exact same 7 locations, guaranteeing a common ancestor. When ALU elements are deposited on top of other ALU elements, the markers become a chronology of development. Very recent human ALU sequences have been used to trace historic and prehistoric human migrations. Since some individuals have new ALU insertions that other individuals lack.